Hi everyone, my name is Nik Jorstad and I'm a scientist in Ed Lean's group at the Allen Institute here in Seattle. Today I'm going to be presenting work from our manuscript titled Evolution of Cellular Diversity in Primary Motor Cortex of Human, Marmoset Monkey, and Mouse, which is currently in review at Nature and was spearheaded by Trygve Bakken at the Allen Institute. And so this project really included contributions from many collaborators across the Brain Initiative Cell Census Network, or the BICCN consortium. And it's really heavily invested in characterizing all the cell types of the human brain with all these different single-cell sequencing-based assays, among other modalities. And so I realize that my audience today is very broad, but hopefully after my talk, you'll have learned some neuroscience and powerful techniques for integrating these single-cell data. So why choose M1, or the primary motor cortex? Well, the primary motor cortex is really an ideal cortical region to study questions about cellular evolution, because unlike some of the other cortical association areas, which have kind of ambiguous homologous structures across species, M1 is very functionally conserved across mammals. And its function is really essentialness for fine motor control. And its cortical geography has also been very thoroughly mapped and characterized in all three species, which is shown here in magenta. So before I dive into the data, I really need to call out all the BICCN collaborators who contributed to this project. This was not necessarily a region that their initial sampling plan called for, but all the groups really pooled their resources and contributed an M1 dataset to this study. So we have single nucleus RNA-seq data for each species, which was generated using 10x with version 3 chemistry. And then on the far left of the chart here, you can see that mouse and human have a layer dissected SmartSeq version 4 dataset as well. For the epigenetic assays, we focused on DNA methylation and ATAC-seq. So for each species, a single nucleus methyl C-seq2 dataset was generated. For ATAC, for human and marmoset specifically, the dual modality SNARE-seq2 assay was used, which is really neat because it captures RNA and ATAC from the same barcoded nuclei. And then for mouse, single nucleus ATAC-seq was used. There's really a concerted effort at the data analysis level between all the groups. So this allowed us to cluster and identify cell types within each technology, and then integrate those cell type calls across modalities and species. So in total, we had something like 500,000 nuclei that were analyzed for this study across all species and modalities. And because my wheelhouse is really focused on the RNA-seq data, today's talk is going to focus on how we identified transcriptomic types within and across species. So here, scientists at the Allen Institute really generated this beautiful pipeline. It's an R-based package for analyzing single-cell RNA-seq data, and the package is called Scratch. Here is the pipeline that we actually used for clustering. It's the Scratch HiCat library, which stands for Hierarchical Iterative Clustering for Analysis of Transcriptomics. And it is what we use to define cell types for this specific paper in the RNA datasets. And all this code can be found on GitHub under Scratch HiCat. Now I'm not going to walk through every step, but essentially the steps are: we QC the nuclei for low-quality cells, donor-specific clusters, and doublets. We then assign cells to a broad type or class, such as the inhibitory neurons or GABAergic neurons, excitatory neurons or glutamatergic neurons, and the non-neuronal cells, which comprise all the glia. And so then we find high variance genes in reduced dimensions with either WGCNA or PCA. These modules are then filtered to exclude ones that tend to correlate with technical noise or technical effects, such as like batch-specific modules or donor-specific modules. We then run a bootstrap clustering and merging pipeline where the clustering module, shown here in red, is done with either Louvain or Ward's clustering, depending on the size of the cluster. And the merging module is then performed, which merges clusters with their nearest neighbor. And they have to meet certain thresholds for like number of cells in a cluster or certain DE gene parameters in order to not be merged. So we ran the HiCat clustering on the human SmartSeq and 10x data, and here on the left I'm showing you the SmartSeq inhibitory clusters that we identified, and on the right are the 10x inhibitory clusters. We got 37 for SmartSeq and 79 for 10x. So one of the really beautiful things about SmartSeq is that it gives us a much better gene detection and over 30-fold more reads per nucleus, but it also costs about 10 times as much, so we can't sample as deeply. And so the beautiful thing about 10x is that you get really broad coverage, but it's at a shallower depth of sequencing. And so the question arises, how can we integrate these relatively sparse and deep data sets with these broad and shallow data sets to find a set of consensus human cell types? And so for integrating across modalities, I really decided to optimize the Seurat-III pipeline. And so after many, many iterations, I found the following to be some really key variables that might help some of you if you're working on this kind of project. And so the first is to start with log CPM matrices instead of the raw reads and UMIs as input into Seurat. I also found that integrating each class with each other, for example, the inhibitory with inhibitory, instead of trying to integrate all the cell classes at once in a single integration, tended to help. And lastly, finding marker genes for each cluster within each independent data set and then using the union of these gene lists as input rather than finding de novo high variance genes seem to really improve detection of especially the rare cell types. So here's the UMAP of the human inhibitory integration, and you can see the red SmartSeq nuclei mix very well with the blue 10x nuclei in the reduced dimension space. So really to identify the consensus types from this integrated space, I performed Louvain clustering within Seurat and shattered the dataset into 225 small but really highly similar clusters. And similar to HiCat clustering, I merged these 225 clusters with their nearest neighbor depending on the cluster size and DE gene thresholds, which resulted in 72 final consensus inhibitory cell types. And so this is really much closer to the 79 that were identified in 10x than it was the 37 that were identified in SmartSeq. So we can see how well our final consensus clusters align with the kind of HiCat specific clusters within each modality using these confusion matrices. So as columns, we have the final consensus clusters, and as rows, we have each modality's HiCat cluster calls. And so we can see that most modality-defined clusters actually align one-to-one with the final taxonomy, especially the 10x, and they have very high Jacquard similarity scores. Now, integration did tend to expand the SmartSeq clusters because you only had 37 of them, and they tended to go to two to three or more consensus clusters. And so one nice sanity check to just make sure you're not scrambling all the biology that's there is to look at how well marker genes are preserved in these new integrated clusters for each modality. So here's a heat map showing all the human inhibitory consensus clusters as columns. The SmartSeq data is on the left, the 10x is on the right, and I found DE genes for each SmartSeq consensus cluster using ROC analysis and am plotting the top 20 genes per cluster here. So we see that the SmartSeq defined marker genes recapitulate very well in the 10x dataset, and this suggests that these consensus clusters are actually biologically relevant. They're not just random clusters that don't share DE genes. They seem to be conserved between SmartSeq and 10x. So I've shown you that the inhibitory neurons integrate very well, but we also perform this similar analysis for excitatory and non-neuronal classes as well. So here's the full human taxonomy across all classes that we identified. And because we have such broad coverage in the 10x data, we're actually able to estimate frequencies and proportions of these cell types, and they're reported here as a bar plot. You can really see that some of the types such as inhibitory cells, have a greater diversity. They have more clusters, but they tend to be rarer than the excitatory neurons, which have fewer clusters but a lot more cells relative to the inhibitory. And so because our SmartSeq data was layer dissected and integrated, we can actually paint the layer metadata onto our taxonomy as well and see which cortical layers these different cell types reside in. And so this really shows that the excitatory or the glutamatergic neurons tend to be more layer restricted compared to the inhibitory. And in this plot, the colors are shown based on their subclass match. So for instance, the purple is kind of the VIP types, the orange is the SST or somatostatin types, and p-valve are the red cells. So again, we really generated these transcriptomic taxonomies for each species. So here are the final cell types for human, marmoset, and mouse. And the main findings here are that we identified about 100 cell types in each species. And based on the coloring, you can see that each species really has comparable diversity of cell types at the subclass level, meaning there's lots of VIP diversity, there's lots of SST diversity, and as you can see in the red, there's about comparable numbers of p-valve cells as well. So how do we go about integrating these taxonomies and finding cross-species homologous cell types? Well, given the success of the human 10x and SmartSeq integration, I really decided to, again, try and optimize the SST07-III pipeline for integration. So some of the most relevant factors that I found for achieving a really meaningful cross-species integration were to first trim the expression matrices to only include genes that are found in all three species. And so in the case of humans, this means going from around 50,000 genes down to 15,000 genes. And fortunately, about 90% of the cell type marker genes tend to be in this conserved gene list for each species, so you're not losing a lot of the identity genes. Next, I found that using Seurat's SC transform normalization really yielded the best results. And so here's the paper that describes that from the Satija lab. But an example of how this fixed things is that one of our marmoset donors had fewer reads and genes detected, and this normalization method really helped to resolve some of the donor-specific clusters. As with the human integration, integrating the data at the class level really helped clusters at a finer resolution than integrating everything all at once. So really kind of integrating a subset of the data tends to be best. And I also found that actually downsampling the data prevented one species from dominating the integrated space. So for example, the mouse had over 50,000 glia while the human had 4,000. And that was based on our sampling strategy, but it can lead to problems in integration. So if you integrate all the glia at once, the mouse data structure is maintained, but really tends to dominate, and so the human data structure gets messy in the integration. So I found that downsampling the data to have roughly the same number of nuclei across species at the subclass level really helped. And so, for instance, for the glial integration, human and mouse both had a few hundred oligodendrocytes in the integration instead of mouse having tens of thousands. And really the last thing that I found that really helped was using these within species cluster marker genes instead of finding de novo high variance genes for anchor finding. And so this really resulted in more one-to-one homologous type alignments, and it also maintained the local structure of the integration better for each species. Also, the really striking thing is that it helps to preserve rare cell types. So if you have just a few, like a few dozen cells out of tens of thousands of cells, if you downsample and really bias towards those marker genes, you can reconstruct those rare cell types in the integrated space. So here's the UMAP for the inhibitory neuron integration across all three species. So each nuclei is colored by the species it came from, and you can see that they tend to mix very well across the whole space. And on the right are the UMAPs for each species, and they're colored by the within species cluster labels. So you can see that the data structure is pretty well maintained, with similar cell types or colors grouping together. And also across all species, you can see that the similar subclasses are grouping together. So you see the red cells tend to group in the same area for p-valve, the orange cells tend to group together for the SST, and so it's nice to see that as well. So how do we identify cross-species cell type taxonomies? That's really the tough question. And so we decided to use a similar approach to how we did the 10x and SmartSeq integration. So we first used Louvain clustering to shatter the integrated space into about 500 small clusters, or we call these metacells. We did this for each class, so here's the inhibitory. And for these metacells, you can see the tree is actually colored based on the proportion of species that contribute to each metacell. So you can see that the deep branches, some of these tend to be very blue, which suggests that they're made up of mostly human and marmoset nuclei. And so we bootstrapped this analysis 100 times, included 95% of the cells in each iteration, and then established a stability score for each branch. And then using a series of these stability and species mixing thresholds, we dynamically prune this tree to establish a cross-species consensus taxonomy. So here's the final pruned inhibitory tree showing the subclasses above and the consensus clusters as the leaves. This whole integration was done with the 10x data, but because we integrated the 10x with the SmartSeq, we can actually link some of the SmartSeq annotations to this tree. So for example, here we can paint the layer dissections from the SmartSeq metadata onto the human and mouse cross-species clusters, and we see that these cross-species cell types actually tend to reside in similar layers as mouse and human, which is really a nice validation of the integration. And another way to visualize the cross-species taxonomy is as a cluster overlap plot. So here we can see the human clusters as rows and the marmoset and mouse as columns. We can see that the species cluster overlap in the integrated space forms these cross-species cell types, which are shown in the blue boxes. So for example, the chandelier cells on the very far bottom right tend to align 1-to-1 across species, whereas just a little up from that, the p-valve 1 neurons are this huge box where there's 10 human clusters contributing to the cross-species type. So here was the final cross-species consensus cluster taxonomy across all of the cell classes. And so this bar plot shows you the number of species clusters that contribute to each cross-species cluster. So some types align very fine at a 1-to-1 or 1-to-2 resolution, while others appear to align many to many and are not as conserved as some of these finer matches. We can also look at how well correlated the average expression of each of these cell types is across species. So here in magenta on this heat map is the Spearman correlation between each pair of species for each cell type. So an example is for the astrocyte 1 type, we can look at the expression comparison over here on the right. You can see that the human and marmoset have higher correlated expression than human and mouse or marmoset and mouse. And globally, you can see across the whole heat map that marmoset and human have more similar expression than either primates do with mouse, which this makes sense based on the evolutionary distance of the species. And also looking at the number of DE genes that distinguish a given cell type between species shows a similar finding. So human and marmoset have fewer DE genes between each other than primates do with mouse. And this is also shown here on the astrocyte plot. Okay, so in summary, the methodology summaries are that cell types can be finely integrated between 10x and SmartSeq, and that the expression of these DE genes tends to be conserved between modalities for these integrated clusters. 2 is that data sets from different species can be integrated at a high resolution. And lastly, in terms of biology, all 3 species tend to have 100 distinct cell types in the primary motor cortex. The cell types reflect evolutionary distance in their DE gene expression, meaning that human and macaque have more similar profiles than either primate does with mouse. And lastly, all 3 species have a highly conserved set of cross-species cell types. So there's around 40 integrated clusters and around total 100 clusters, so about 50% conservation. And with that, I'd like to thank you all for listening. I'd like to thank the BICCN for sponsoring all this work and all the collaborators who took part of this massive study, all the analysis that was done by these many different labs, and lastly, our founder, Paul Allen, for his vision and support of these efforts as well. Thank you.